Welcome back to the Soundbites podcast here at the ASGCT event in Boston. Very excited to interview a panel of different people doing some really powerful research that is focused around an award, Jennifer, that you just won. So Jennifer Adair, can you please tell us a little bit more about the award you just won?
Hi, thanks so much for inviting me to be here, Ben. So today I'll be receiving the Award for Excellence in Diversity, Equity, and Inclusion. And as I'll say in the remarks to that award, this is really about making a bigger table that can fit more diverse perspectives so that we can ensure cell and gene therapy gets to every patient who needs it when they need it, no matter where they live in the world. And so I'm super excited to have a couple members of my team here today, as well as two visiting scientists from Uganda who are part of an active NIH partnership with us to develop capacity for cell and gene therapy towards the treatment of HIV.
Excellent. Fantastic. All right, we will start with the first person. George, can you explain yourself and your role?
Hi everyone, thank you all for inviting me to be part of this podcast. I appreciate the opportunity. My name is George Ssenyange. I'm a research scientist based at the Joint Clinical Research Center in Uganda. I mostly do HIV research, and my research centers around the problem that's currently the main barrier with HIV cure, trying to eradicate the latent reservoir. So in my research, I'm proposing a strategy to engineer gene circuits that are programmable so that we can sense cells that are activated, and then the cell can be programmed to kill itself. So if this works, I think it can be a new paradigm in terms of HIV cure research that could help us in combination with other therapies that are being made up to try and cure HIV for good.
Very interesting. Thank you very much. All right. Next candidate.
Hi, everyone. Thank you very much for inviting me to be part of this podcast. I'm Gertrude Kiwanuka from Barra University of Science and Technology in Uganda. I'm very grateful that I've been given the opportunity to be present in this ASGCT 2026. I'm part of a group in Uganda that is developing a training program funded by the NIH to train researchers in cell and gene therapy for HIV cure. But at the same time, I also have keen interest in research ethics. I am leading a training program back in Uganda that is developing and building research ethics capacity. So I have enjoyed the meeting here in Boston, interacting with scientists, clinicians, listening to manufacturers, and I am taking it as an incredible platform that is giving me so much information, so much collaborations, and I have so much to take back home as an advocate for cell and gene therapy. Thank you.
Fantastic. Yeah, it's a fire hose of information here at ASGCT. All right, on to Rachel.
Hi, thank you for having me. My name is Rachel Cunningham, and this is my first ASGCT as a PhD. I'm a postdoc in the Adair lab. Our research focuses on engineering and transplant of hematopoietic stem cells, and in particular, my project is trying to remove some of the barriers involved in the patient impact of chemotherapy and radiation conditioning. And there's a lot of research going on in that particular field at ASGCT this year, and it has just been so cool to see all these different groups making advances in this area. I'm very grateful to be here, and it's a super cool time.
Excellent. And last but not least.
Hello, my name is Motahareh Arjomandnejad, and I'm a postdoc in Dr. Adair's lab. Similar to Rachel, this is also my first time here as a postdoc, which has made a different experience for me. My project focuses on, we previously developed a AAV capsid that has a skin targeting ability, and we are in Dr. Adair's lab. We are trying to actually utilize this capsid to target genetic skin disorders. For skin diseases, unfortunately, there hasn't been a lot of talks in ASGCT. However, there are a lot of biology that we need to actually have a better understanding of, and ASGCT have been a great place to actually educate me towards that direction.
Fantastic. Thank you to each one of you.